A very good morning and good afternoon to one and all present. On behalf of Preventive team, I would like to thank all the viewers and a special thank to all the esteemed speakers who took out time from their busy schedules and agreed to be part of this event. This workshop is being conducted as part of the Preventive project. As you can see the logo on the extreme left, and the project name is Risk Management and Prevention of Antibiotics Resistance, which is being co-funded by the Erasmus Plus program of the European Union. And you can see my photograph on the right, Dr. Kiranjit Kaur. I hope you can see my cursor. And I'm the only one who is going to introduce myself. So I am basically a microbiologist. And right now I'm working as a coordinator for this project. And for today, I'm also the moderator and the speaker for this workshop. So before I begin, I would like to mention that all the speakers have been requested to use as layman language as possible, because we know we will be having, the audience from multiple backgrounds and from different streams. So please don't worry if you don't understand the title and there are a few words which are going, over the head. We will try to make it as understandable as possible. Okay. So once you have attended this workshop, it will take around a couple of hours from your time. You will be able to explain how the spread of antibiotics or antibiotic resistance bacteria happens in food chain. Secondly, you will also gain knowledge how to follow the food and safety guidelines in preventing these foodborne infections. This workshop is being conducted on Zoom and it is being streamed live on YouTube. I'm sure when you listen to so many experts, as you can see on the flyer, which has already been circulated to you from your seniors, you can see there are many questions which will pop into your mind. So please don't worry. We will try to answer as many questions as possible in the limited time. So whenever you have any question in your mind, please use chat box of the YouTube. Just type #query and preferably type your name and write the question. In case you have missed out on this workshop or you miss out on any session, do not worry. The content of this video will be downloaded and will be uploaded on the official website of Preventive and other social media platforms like YouTube, Facebook, Twitter, and LinkedIn. One of the important things, I think, because of which many students will be joining this workshop is e-certificate. So do not worry, we are going to share the link of the e-certificate for the e-certificate by the end of this workshop. So you have to be there because there may be some questions that you need to answer before you get your certificate. Okay, so be there. We will do the needful by the end of the session. All right, so this is the brief agenda. I'll not go into details of it right now. I'll just read the highlights. We are going to have an opening session by me, that is Dr. Kiranjit Kaur. Hello. And then we have the following session by different experts, and I'll be introducing them whenever they have their sessions. So the first section will be talking about how antibiotics and food is related. And then we have the talk by me again. I'm sorry, there will be too much of me in today's session, so you have to bear with me. Hope you will like me. Then the section by me will be covering how and which are the food products wherein we are having this problem of antibiotics and antibiotic resistant bacteria. Following which we have a session from EFSA, European Food Safety Authority, a very famous organization of Europe based on Italy, who will, the session will be covering about how the European Commission or European Union, they are taking care of the risks involved in antibiotic resistance and how they are taking steps to break this chain. And last but not the least is a debate session wherein we have learned about the problems. Like by this time, we would have learned about the problems. And in the end session, we will also learn how to break this chain, that we also talk about the solutions. And do not worry, I've not missed intentionally on this break session. We are going to have a break session in between as well. All right. So since this is an overview session, I'm going to start my talk right now. This is the first slide from my section, which is an overview. So I'll try to be as brief as possible and as simple as possible so that I set a basic background for all of you. And I need your attention, obviously, for all the sessions, but specifically for this section, because if you do not understand the basics, why this workshop is important and why you are being involved in this workshop, then you may not find the other lectures interesting. So please try to understand why this workshop is important. All right. Okay, so I would like to start with a very simple and basic questions. Obviously, you do not have option to give answers, but I can give answer on your behalf. Why do we eat food? So food is something, which connects all of us, and it is something which is being consumed by all the living beings. So obviously it has many aspects to it, but I've just highlighted few of them. It keeps you live longer. It keeps your skin glowing. It keeps your teeth and eyes healthy. Blah blah blah. So you talk about anything. I'm talking right now. My eyes are blinking. I have my mind, working on this presentation. My hands are moving. Everything is dependent on food, right? And how does this happen? Because this food is rich of carbohydrates, fatty acids, vitamins, minerals, proteins, many things. And all these things, they provide energy to our cells, right? That is why we are able to do whatever we are doing right now. So this food is very important. This all of us agree. But do you think this food may also contain something which is harmful to us? Do you even believe it? Or have you ever, I don't know whether you had any thought before this workshop title had come to you, that is an alarming situation. Why we are talking about food as an alarming situation? So let's try to understand that. Okay, so this food, as I told, it is very, very useful, but it may also contain antibiotic residues or antibiotic resistant bacteria. Again, I'm repeating, do not worry if you don't know these terms. I'll try to explain in a minute or so. So basically, these antibiotics are nothing, but these are the medicines which are used to kill or inhibit the growth of bacteria. Bacteria, I'm sure all of you must be knowing, these are small organisms, okay, which are present everywhere, which cannot be seen by naked eye. So if I use today the word COVID-19, I'm sure everybody knows about COVID-19, but that's a virus. So it's you can say a distant relative of that virus, but they are named as bacteria. They are little bigger in size as compared to viruses, but still smaller that we cannot see them with naked eye. All right. So please keep this point in mind, which is very, very important, that the bacteria are present everywhere, whether it is soil, human environment, inside us, outside us, in water, in animals, everywhere. And they can also spread from one place to another. Don't worry, I'll come back to food. Why I'm talking about bacteria, you will come to know in a minute or so. All right. So these bacteria, they are divided into two categories, good bugs and bad bugs. So mostly the good news is that most of the bacteria are good bugs. But the problem is with the bad bugs. These bad bugs, they cause diseases. All right. And I think I need not to explain the term disease, which is very, very common. So what do? Do we sit back if we have any disease? No. Luckily, we have some medicines which are named as antibiotics, and they help us to fight these infections. As you can see in this GIF image, I hope it is visible and working on all the systems from wherever you have logged in. So you can see this person, this yellow and blue person who is having that getup of a fighter. That is termed as an antibiotic, that is symbolized as an antibiotic in this GIF. And these are the bad bacteria. So antibiotics is able to fight against these bacteria. So whenever we have any disease, these antibiotics can fight with them. However, these antibiotics are not only useful in humans, as I already told, that these bacteria, bad bugs, they can be anywhere. Okay, so they can also cause diseases anywhere. Anywhere means to all the living beings. So those also includes plants and animals, right? So whenever these bacteria, these bad bugs, they also cause diseases in these animals and plants, we have antibiotics which can take care of their infections as well, right? So now here comes the term food. We all eat plants, right? We all, not all of us, but most of us who are non-vegetarian, they also eat the meat. But all of us do have milk until unless you have some allergies or other things. Please take care of yourself. So we all consume different dairy products which are there from animals. And why I'm talking and why I'm focusing on that, because just for your information, out of 10 antibiotics, 7 antibiotics or more than that are used in animals and plants compared to humans. And the important point here is this will connect both food and superbugs and antibiotics. Why we are talking all of this? Because when animals consume antibiotics, more than 50% of the antibiotics, they remain unmetabolized. Unmetabolized means they remain unused in the animal system, and they are being excreted out as such. So now what happens? What happened with the passage of time when we were using antibiotics in humans, animals, plants, everywhere? So what happened? Slowly and steadily, these antibiotics were used by humans, were used by animals, and they were excreted out into system. As I've already told, they are not being metabolized completely. So they enter into water channels, they enter as manure from animal side, and then they enter into the environment. And our food and plants, they directly get a bad impact by these antibiotics. So I think now you understand how antibiotics, antibiotic resistant bacteria, and how this food, they all are interlinked, right? So since bacteria are everywhere, the infections are everywhere, and antibiotics are everywhere, so all this is interlinked. So now what happened? Everything was going happy-go-lucky. Like, it's okay if we have an infection, we have an antibiotic. There's no problem, right? But the bacteria are also not sitting idle. They may not sit back and just say, okay, antibiotic will come, let it kill me. No, they also became smarter and smarter by time. And they followed the theory survival of the fittest, right? Which is very, very common. That holds true for us as well. Same is the case what we are seeing with COVID-19 nowadays. I'm sure all of you must have heard about the variants. No? Now Delta variant is there. Now Gamma variant is there. Beta, I don't know. You name a category and you have that version of COVID-19. So what is that? COVID-19 virus, it is trying to change itself so that it can keep on spreading itself. So same thing happened for bacteria. They thought one day, yes, this is the time of revolution. Either we will evolve or either we will die. But the sad part is they evolved, right? So this earlier situation wherein antibiotics were efficient enough to kill these bad bugs, today's situation is that one bacteria can be killed, second can be killed, but the third one has become strong enough, and now the antibiotics are not effective. So whenever we are consuming these food products who are having these bad bacteria, we will have bad infections. And if we eat antibiotics, we will not be able to get rid of that infection. I hope I am able to make myself clear. So just for your understanding, a situation where antibiotic is no longer effective against bacteria, that is known as antibiotic resistance. And we must understand that effectiveness of antibiotics is not going down, but the bacteria is becoming smarter and is becoming more stronger. And that is why it is termed as antibiotic resistant bacteria or superbug. All right. So why are we talking about it? The key problem is, one thing is that all of us consume food. So today's workshop, since it is focused on food, so I'm going to relate everything in terms of food. So all of us are consuming food. All of us are prone of having infections from food. So this antibiotic resistance, it can happen to anyone of any age in any country, right? Plus, recently, I think a couple of years back, WHO, the World Health Organization, has also included this as one of the top 10 threats, which is affecting our global health. And it is being estimated that by the year 2050, 10 million deaths will be due to this antibiotic resistance problem. All right. So what will be the impact? If we have so many infections around us, what will happen? We will have longer stays at home, at hospital. We will have more cost of antibiotics, because if one antibiotic is not working, we have to go for another antibiotic, right? And that will be more costlier. There will be high bills. There will be loss of earnings. And like today, we are having less travel because of COVID-19. Sometime down the line, we may also have less travel or maybe no travel at all because of this foodborne infections, which are due to these bad bugs. All right, so don't worry. We do have solutions, and one of the easiest and one of the key solutions is, educating yourselves and creating awareness amongst your group and your loved ones, so that we can help each other. At least, in case of COVID-19, we were not aware that, okay, it happened suddenly, although we had some versions of it earlier. But COVID-19, it globally impacted. It was a pandemic, so we were not aware, and we were not planned. We didn't knew what to do in the beginning. Today we are aware of it, but about this antibiotic resistance, we already know that this problem is there. Then why should not we act, and why should not we educate ourselves for this, about this problem? All right. So keeping this in mind, this agenda has been designed for today's workshop, and I hope I set the background nice, and I hope all of you now know why this workshop is very, very important. So I would now request our next speaker, Dr. Himanshu, to please start sharing his screen. And by the time, I would use this opportunity to introduce you to our audience. So, Mr. Himanshu Shekhar Pradhan, sir, can you hear me?
Yes, please.
Yeah, perfect. So can you please turn on your camera so that our audience can see your, yeah, thank you. Thank you so much. So you can also start. Yeah, great, sir. We can see your slides. Okay, so perfect, sir. You can also put it on slideshow so that it's clearly visible to the audience. Great, thank you so much. Okay, so before I hand it over to Mr. Himanshu Shekhar Pradhan, I would like to introduce and give a brief background about him. So he is a public health management professional having more than 17 years of experience. He had a chance to work with many reputed organizations such as UN agencies, United Nations agencies, World Health Organization, United Nations Development Program, and United Nations Population Fund. Currently, he's working as an associate professor at Kalinga Institute of Technology, deemed to be university, that is since 2017. Dr. Himanshu has a good track record of facilitating and managing innovative projects, and Prevented is also one of the projects. He's also a lead coordinator from Kalinga Institute. Not only this, antibiotic resistance, he also has his experience in multiple domains, such as HIV/AIDS, nutrition, migration, strengthening health information system, health action in crisis, public health in emergencies, so on and so forth. There's a long list, and keeping time in mind, I cannot keep on talking about it. So I hand over to Mr. Himanshu to kindly proceed with the lecture. Thank you so much.
Thank you, Dr. Kiranjit, for a humble introduction. So I respected all the dignitaries, all the participants, and I welcome once again to this forum. So I'm very thankful to Dr. Kiranjit. She already set the context of this today's deliberations, and I will be briefly discuss antibiotics and food. So what are the links between antibiotics and food, and how antibiotics are coming to our food chain and it's creating problems? So my presentation is broadly divided into two parts. And first part, I will discuss the role of food in health and infections. And second part, we'll be discussing how antibiotics and antibiotic residues or antibiotic resistance bacteria enter into the food chains. So Dr. Kiranjit already, she highlighted how food is very much important for our health and well-being. Because this is everybody knows that food can be defined as anything solid, semi-solid, liquid, which when swallowed, digested, or assimilated in body provides it with essential substance called nutrients and keep it well. So food is the basic necessity of life. It not only keeps a person alive, but also provide energy for growth and development. Food provides our body with what they need to stay alive, be active, move and work, stay healthy, and heal themselves, prevent and fight infections. Broadly, function of food can be divided into three areas. One is physiological functions, second, social functions, third is psychological functions. So physiological function basically is very much linked to our health and well-being. So food provides energy to the body, what Dr. Kiranjit also highlighted. It also facilitates growth and development. It enhances muscle strength, bones and teeth, keeps our body warm, and keep our body healthy by helping our immune system to prevent or fight disease. It also facilitates repair and healing the injuries, helps our brain to grow, function properly, and development and proper working of our key vital organ of the body. So that's why food is very much important for life, for health, and protecting ourselves from infections. As we understand, there are various nutrients, for example, like carbohydrate and fats are a rich source of food which give the energy to the body, proteins and minerals, and they facilitate muscle development. Essential fatty acids, protein, minerals, they facilitate some regulatory functions and maintenance of body temperatures, activation of enzymes. Proteins, minerals also facilitate repairing the cells and tissues, and vitamins and minerals, they are protective foods. They are facilitator, giving the strength for injuries, enhancing, protecting ourselves against injury and diseases. So now we are quite clear that food has very powerful function to maintain our health and well-being. But at the same time, what Dr. Kiranjit already highlighted, because if that food become contaminated, or if the food create problem for us, then it is detrimental for our health and well-being. So access to sufficient amount of safe and nutritious food is key to sustain life and promote good health. However, use of food containing harmful bacteria, virus, parasites, and chemical substances cause more than 200 diseases, ranging from diarrhea to cancer, as per WHO's. Unsafe food creates a vicious cycle of disease and malnutrition, particularly affecting infants, young children, elderly, and sick. Foodborne illness are usually infectious or toxic in nature and caused by various factors, starting from bacteria, viruses, and chemical substances. One such chemical substance that already discussed, setting the context by Dr. Kiranjit, called antibiotics. So abundant use of antibiotics leads to antibiotic residue in frequently consumed food. That is a matter of concern. Residual antibiotics in food, it has adverse effect on human directly by causing disease, where low doses of exposure is regularly being given to the animals or anything, and also it indirectly harm by antibiotic resistance. So in subsequent slide, we'll discuss in detail how it happens. A residue of antimicrobials in food have received much attention in recent years because of growing food safety and public health concern. And the major public health significance of antibiotic residue include development of antimicrobial drug resistance, hypersensitivity reactions, carcinogenicity, mutagenicity, and teratogenicity, where the embryos and other things get affected due to prolonged antibiotic use and disruption of normal intestinal flora. So that's why antibiotics and health is a very closely linked, and how it is. But next question is that how these antibiotics enter to our system. So even though there are another aspect, Dr. Kiranjit already highlighted, antibiotic resistance, the infection in humans can cause long-term illness, increase the frequency of hospitalizations and treatment failure that can result into death. So now let's understand how this antibiotics in food, how it comes to food and how it entered. So the second part of my discussion, I will focus that how antibiotics and antibiotic resistance bacteria spread in food chains. So before understanding the spread of food chain, let's understand where are the places where we are using antibiotics more. So one such huge use of antibiotics is called food animals. Use of antibiotics in food animals. So what are the food animals? Food animals are the animals that are raised or breed to produce food for human consumption, such as eggs, meat, milks, and etc. So there's a wide range of, this cattle, these goats, the pigs, chickens, dogs. So there are different, which is predominantly used by human. Now there's a growing demand for food of animal origin across the world. So that's why volume of antibiotics used in animal is continuing to increase the worldwide due to this demand. And also often it produced through, which leads to intensive animal husbandries. But the irony is that overuse and misuse of the antibiotics in animals and humans is contributing to rising threat of antibiotic resistance. So there are 4 broad areas where these antibiotics are predominantly using in food animals. What are those? One is therapeutic purpose. That means we are treating disease when animal falls sick. Prophylactic purpose. That means facilitate, prevent the animals, those who are risk of being infected. Metaphyletic. These are the situations where if any outbreak happen in any animal herd kind of things, we treat those who are clinically healthy animal so that they will not get affected. And the last one is growth promotions. We use antibiotics for giving boosting the weight of the animal, quick speed up the growth process. In case of pigs and chickens, this predominantly also used. But the gravity of the situation, if you see, as for this economic cooperation and development estimates, in 2010, across the globally, around 63 tons of antibiotics were used for animal sectors, food animal sector, and which is projected it will be reaching to 105,596 ton by 2030. So that is a gross 67% increase of this use of antibiotics. And if you see the top 5 countries across the world which are using these antibiotics predominantly in food animals, that's China, US, Brazil, India, and Germany, where they consume huge antibiotics for food animal sectors. And also another way, because already the poultry also we use huge antibiotics in the poultry, because if you see the poultry, everybody, majority people across the world, they use poultry. So poultry industry use antibiotics to improve meat production through increased, to enhance the growth promotions and disease prevention. And it is growing use of antibiotics that has resulted in increased concern over the development of antibiotic resistance in poultry system. Another areas where much antibiotics also used, it is called as aquatic or aquacultures. Globally, aquaculture contributes 8% of animal protein intake in a human diet. And with rapid growth and demand for aquaculture, industry faces challenges in form of diseases. Any disease to this species might result in loss of complete culture. That's why, so for preventive measures, farmers started using antibiotics and found good result. And if you see the Indian situations, aquaculture constituted over 1/3 of India's total fish productions. But if you see this magnitude or the gravity of the volume of antibiotics used, as per 2017, 10,259 tons was using were used for this aquaculture, which is projected by 2030, 13,600 tons. So that this giving an idea as to us that what volume of antibiotics are being used for food animals. Another area where also antibiotics used, it's called crops. Antibiotics are also used for crops through amount, though the amount is relatively low in compared to quantity used in livestock, but still it is estimated 0.2 to 0.4% of total agriculture antibiotic consumption. Antibiotics and antifungals are used as pesticide to manage crop disease. And this may spread of development and spread of resistant germs by contaminating the surrounding soils and water. So now what we are trying to understand for last 2 minutes is telling that there's a gravity of the situations and antibiotics are used across, be it crops, be it food animals, be it poultry, be it aquaculture. So that means everywhere we are using antibiotics. But the question arises, even though we are using, what are the mechanisms, how these antibiotics are entering to the food chains? So let's have a look to these aspects. So spread of antibiotic and antibiotic resistance through food animals. Because as Dr. Kiranjit already discussed in previous deliberations, many classes of antimicrobials that are used for humans, same antibiotics also used for food animals. And such, because since from a previous slide, we have seen that the volume of antibiotics, because the matter of the fact is that due to mis or indiscriminate use of antibiotics, which accelerate the development of antibiotic resistant bacteria and which escape and spread into the communities. And there are two ways this antibiotic resistant bacteria or antibiotics resistant germs, it enter or reaches to human. One is directly, another is indirectly. Indirectly, along with the food chain for consumption of contaminated food or food derivative products, and directly by direct contact of infected animals or a biological substance like blood, urine, feces, saliva, or semen. So these are the two pathways. Directly or indirectly, antibiotic resistance food and bacteria or antibiotic related stuffs come into the human being. If you see this, a diagram here, we are telling that antibiotics. This is a diagram is showing that how antibiotics transmitting from animals to human. If you see the antibiotics given to animal, then it has one option to once it reaches to animal, either it release the antibiotic residue or it also release the resistant bacteria which are constantly coming in contact with the antibiotic, and then they become resistant. So they have two options: either the antibiotic residue or antibiotic resistant bacteria or superbugs, either it enter to environment in the form of waste or animal products, or it has another options. It will go to go as a waste, as a animal waste, as a manure in planting or plant farming. So in both ways, either it is as a plant animal product or as a waste, it reaches to human in the forms of animal product. Whatever we can meat, milk, egg, or whatever, and another vegetable and fruits. And if we do not maintain proper hygienicity kind of things, and it will ultimately lead to human transfer to human being. So let's take a few examples that to understand how antibiotic residue or antibiotic resistant genes in passing to reach to food chain and ultimately leads towards. Take an example called milk, antibiotics in milk. Because milk and milk products present 10% of food expenditure in India. Why antibiotics may be found in milk? Because there are 4 reasons which leads that antibiotics may be found in milk. One is called mastitis. It's a disease in cattle. Another is when we are using antibiotics for managing the disease. Also, antibiotic used for growth promoters, even though it is restricted in many countries, and contamination of food and water and fodder from environment and reaches to cattle and ultimately comes. So let's see how it happens. Because before that, I just wanted to let you know, what is mastitis? Mastitis is one form of common clinical presentations which encountered in dairy farms and milking cows, buffaloes, affected disease, and which have shown contain bacteria and with a wide spectrum of resistance against the commonly used antibiotics. In some cases, multiple drug-resistant bacteria have also seen to co-infect animals suffering from mastitis. And most important thing, another concept what is called a withdrawal period. Withdrawal period means it is a concept because it is a time between administering of antibiotics and deriving a product. Suppose now I am administering antibiotics and immediately I will milking the cows. Or suppose now I am administering antibiotics and within very shortly I am using the meat. So during that period is called it's called withdrawal period. There is a certain period. During that period, we should not use any animal product to whom antibiotic is administered. But due to economical issues, the farmers they may not wait for a long time. So immediately and maybe due to many other not good farming practices, we quickly use milk product or animal product, even though quickly before immediately after administering antibiotics. So during the process, the antibiotics and antibiotic residue are entering into the food chain. If you see this slide I have taken this slide from Dr. Professor Alfonso Giacconi because sometimes he has also given it a research. So this slide is depicting how that antibiotic residue entering into the milk. If you see that there's a growth promoters because antibiotics are using as growth promoters and antibiotics are releasing to environment through fodders and other things. It is coming to cows. We are also using antibiotics for treating the mastitis. So during the process, this milk getting contaminated and milk having the residue, because if you do not maintain the withdrawal period, the milk having the antibiotic residue if you milking the cows quickly, then the residue comes. And at the same time also, some of the pathogen resistant bacteria, they go out fecal material and come to the soil. So both ways, either the persons or as a human being, if we use that milk having residue antibiotics, or we get contact with the pathogens environment, then we get affected with either antibiotic residue in our body because we are consuming the milk having antibiotic residue, and prolonged time of antibiotic residue milk, if you consume, our bacteria inside our body also they become resistant, and also the other resistant pathogens which are also coming to our body, so that the antibiotic resistance it will happen, and this resistant bacteria also we are getting. So this is a pathway through which milk antibiotic residue and antibiotic resistance bacteria are entering into the food chain. If you take another example, let's take an example that how spread of resistance of bacteria from poultry. If you see this diagram, antibiotic resistance bacteria from poultry, it can reaches to human or food chain. There are three ways. One, via direct contact with farmers. Because if you see the farmers standing here, either the farmers they are directly slaughtering, or directly they are preparing the meat and all sorts of things, eggs. So there are directly antibiotics. Resistance bacteria. It may come directly to the persons who are handling, or the farmers to the handling, or it may come via food. It may come via contaminated food, or the excreta of the birds or this poultry. It comes to the environment, and this excreta it may reaches to water bodies or in the environment, and which also come getting contaminated water, contaminated soil. It also comes to the human being. So there are three ways that thing is ultimately reached to human being. So this is a pathway, because even though it is in poultry and in food chains, and there are different other mechanisms, it reaches to human being. So resistant bacteria may spread from general, and once it reaches to one person, and there's a high chances that this bacteria also they spread from one person to another. This is a second example. We wanted to understand how the resistant bacteria and poultry, resistant bacteria and entering from poultry entering to the food chains. If you see another example that we try to understand here, how the meat, because if you see this this diagram, it is reflecting that antibiotics, when you are administering antibiotics to animals, to poultry, so antibiotics kills susceptible bacteria, but there will be some bacteria they become resistant, and this resistant bacteria they grow and multiply. So and ultimately it comes to either animal meat, or it comes to what is called eggs, or some of the bacteria it also released to the environment, and resistant bacteria it environment and contaminate soil and plants. And if this contaminated soil, maybe fruits, vegetable, eggs, or meat, and if you do not maintain proper hygienicity, inadequate cooked food, or when the raw materials are contaminated or gross contaminated with other food environment during the preparation, it leads to antibiotics. There's a high chances that antibiotic resistance bacteria or antibiotics related stuff enter into the food chain and ultimately leading to us. Another example, I'm just trying to reflect it here, how aquaculture also facilitating a role that how antibiotic resistance bacteria is also entering into the food chain. You see, there are 70 to 80% of antibiotics end up in the environment. Whatever antibiotics used in aquaculture, it leads to environment 70 to 80%. And use of unapproved antibiotics in the integrated practice of poultry and aquaculture increase the chance of spread of antibiotic resistance from one system to another system. One consequence of use of antibiotics in aquaculture is the presence of residue of drugs in the edible tissue of the treated fish, as well as it percolate down to the environment, and which is also ultimately leading or reaching to human being. Another example, if you see that how antibiotics enter into environment, because if you see that in this diagram, we are trying to explain various aspects, because lots of antibiotics are being used in what is called, in healthcare facilities, hospitals and other things. And these antibiotics, if they are not properly, what is called as during the process of processing, effluent processing kind of thing, waste properly not being processed, that antibiotics many times and directly come into the water bodies or environment. And the other thing also we discussed, due to animal husbandry, plant products, aquaculture, of some of the antibiotics residue and antibiotics resistant bacteria that also come directly to environment, so that it's contaminated water bodies and environment. And another important aspect is pharmaceutical industry, because pharmaceutical industry, they are effluent coming out of the pharmaceutical industries. If they are not properly, what is called as processed, then the antibiotic residue also enter into the water bodies. And also many times accidental or intentional release of antibiotics from the production plant come into the environment. Because once it's come to the environment, it is come into the water bodies and environment. And in this, if this water, contaminated water, if we will use, so definitely we will get a human being, animals will also directly get that antibiotic residue or resistant genes. So these are different ways that antibiotics resistant genes and antibiotics are entering into the food chain. So to summarize, another important, the CDC has given another very, important fascinatingly, that how, you see that first line, it is showing that antibiotics are given to the animals and on all the products, animal pigs or whatever, this poultry, and animal kills and antibiotics kills most of the bacteria, but resistant bacteria survive and multiply. And if this bacteria either it reaches to us, already I discussed, either through animal products, or it may go to directly contaminated to the environment, and through that environment, either it can come to water bodies, or that water contaminated environment, soil and water, it come contact with the vegetables. And sometimes also this contaminated bacteria and contaminate our bacteria, resistant bacteria, it will come to our kitchens due to cross transmissions. And also, fecal material of this poultry and animals, which the contents, these resistant bacteria or residues, it may also call to the environment, so that it spread their things. And once this spread happen, and once these things come to our food matters and all sorts of things, so we very quickly, human being get exposed. Through the, either through drink, either through food material get exposed, or through directly vegetables and other things. So once we expose it, now we are in a receiving end, which creates making a very much vulnerable situation for human being for getting either more residue of antibiotics we are coming to us, or we are getting resistant bacteria. So over the period of time, we are becoming very much vulnerable due to antibiotic resistance, prolonging hospital stay, difficult to treat disease, and which create a lots of difficult situation to handle. So this is my a brief reflection how antibiotics and antibiotic resistant bacteria enter into the food chain and ultimately leading to us and create a problem, what is called antibiotic resistance. And I believe the my next presenter, definitely she will highlight, because I have not given any statistics, keeping in view, this next presenter she will highlight about the magnitude, what is the prevalence of different antibiotics related issues among different animals and food products. So thank you so much for your patientful hearing.
Thank you so much. Thank you so much, sir, for explaining in a very simple manner as expected. I am sure if I would have missed out on some point, you have made it very, very clear to the audience. And I think by now they know what are antibiotics and how it is related to food chain, and how, why this workshop is very, very important for everyone to attend. Thank you so much, sir. Now I would like to share my screen. And so can you just stop sharing the screen so that I can. Thank you so much. Great. So I know, so we also have a couple of questions, but keeping time in mind, since we are already running a little late, so I will use that. I will take those questions maybe after or before the break, so that we complete at least the problem section in one go. All right. So I will quickly move towards my presentation, and since we know that this presentation can just give this presentation. I hope it is visible. Okay. So basically, this presentation, I'm going to talk about the epidemiology of resistant bacteria. Sir has clearly already have shown, he has talked about multiple aspects. So in order to save a little bit of time, this presentation is basically about the percentages and the numbers. So you don't have to write down or pen down anything. You just need to be little aware. This presentation is only to make you understand that, where are we in today's scenario. All right. So just to let you know, epidemiology is a term which is basically termed as is used to define the spread and control of disease. So this is a technical term. I'm not going into details of this. You can read it. We can share the PowerPoints with you later on. All right. So who does this, experiments, and who make us understand what is the surveillance? This sir has already covered, so I'm going to skip it. But just to let you know, at national level, that is at Indian scenario, the two main organizations that are taking part in it, that is ICMR and DBT. And most of the data that I'm going to share today is based on three major organizations. That is REACT, that is a website which works on antibiotic resistance very actively, ICMR, and CDEP. So as sir has already highlighted, I'm going to discuss the percentages or number here, but just I could not miss out on this slide that this is one of the report which is being given by ICMR. Okay. And if you see this pie chart, only 3% of studies, if you see, are, we only have 3% of studies which have tried till today in Indian scenario to understand what is the presence of, how much is the presence of antibiotics or antibiotic resistant bacteria. So you can very well imagine, compared to human data, it is very, very less, but still I've tried to manage some data. So I'll quickly share that with all of you. This I'm going to skip, has already been covered by sir. These one also. All right. So this is one of the organization, as I've already mentioned, CDEP, which is working very actively and in this direction. So if you go on the website, I intentionally added this slide so that in case you also want to know where our countries stand and where the database of other countries stand, you can always click on projects and click on resistance map. And then you have an option of choosing which countries you want to compare, and you want to see the data of which organism. So here in this slide, I've just tried to show that this is the pattern of antibiotic resistance of Escherichia coli. This is one of the organisms which is responsible for multiple diseases, but is also responsible for foodborne disease, specifically diarrhea. So if you see in Indian scenario, you see all the bars which are raised compared to European countries, especially Italy and Netherlands. And for sure, there must be something which is being applied in these European countries, or there are some rules which are being strictly followed in these countries which are not being followed here, irrespective of the fact that we have more population, but we have all the bars raised. So trust me, you can go to see their website and check for different organisms. I'm sure more than 80% for them we have, bars little raised, which is not a very, very good thing. All right, these facts again has already been shared by Dr. Himanshu, so I'm going to move forward. All right, so I added few, researches here just to make you understand about the percentages. Although the studies are very less, as I've already shown, but still you can see that in one of the studies in Kerala, it was found that out of all the raw milk samples that were being collected, 10.23% of raw milk samples had antibiotic residues, and that antibiotic name was oxytetracycline, and this is also being used in humans. In another study, it was being found that out of 492 samples, 16% were found to be positive for antibiotic residues, and the study is from Punjab. This is one of the important studies wherein I would like to have your attention. In one of the studies in Karnataka, an effort was made to understand that if we heat the milk sample, then do we have the less impact of antibiotics? So it was found that yes, temperature does play an important role, but not completely. So it's not that if you have heated the milk, at a very good temperature or at the prescribed temperature, that milk is still not free of antibiotic. All right. This is another study again done in Indian scenario near the places of Hyderabad, and it was being found that, like Dr. Himanshu mentioned, that we have pharmaceutical industries which keep on, putting all these antibiotic residues or waste materials into the rivers and all. So it was being found that near city of Hyderabad, all mostly the lakes which are near the pharmaceutical companies, they all had good amount of antibiotics, and these water bodies are also being used as a source of water to some of the people belonging to rural areas. All right, just to give you a quick overview about the poultry in terms of antibiotic resistant bacteria. In one of the studies, it was found, and this study was conducted in multiple places of India, like Uttar Pradesh, Haryana, Rajasthan, Punjab, wherein in total 217 bacterial isolates were found. And sadly, all the strains of E. coli, again, I've told E. coli, the organism which is responsible for diarrheal infections, was shown resistant profile of 100%. Klebsiella pneumoniae showed resistance profile of 92%, and S. lentis showed the resistance profile of 78%. So you can imagine, even if we have the less data, but the numbers are quite high and alarming. This is a screenshot which I've just taken for your information for ICMR, the report which I showed you in the very beginning of this session. So you can always again go to that ICMR website and have a look on the database. These are some studies in context of livestock. Again, I'm not going into details, but you can have a look, quick look on these on the extreme right. I hope you can see my cursor. There are different percentages of the resistance to antibiotics seen in the milk samples, be it a raw milk sample, be it a cooked milks, the heated milk sample. We still have some bacteria which are showing the resistance profile. All right. In context of aquaculture, again, there was one study wherein especially in Kerala, which are rich in the seafood like shrimp, shellfish, and clams. Frankly speaking, I don't even know what clams are, but anyways, I presume that they are also seafoods. So these vibrios, they were being taken or isolated from these aquaculture organisms, and it was found that all the species sadly were resistant to ampicillin, which is one of the very common antibiotic. And here also, if you can see, another antibiotic, ceftazidime, the resistance profile was 67%. So this is really sad. Similarly, in another study from urban lakes, it was found in this one of the common fish. I don't know, maybe the non-vegetarians must be knowing, and must also have some saliva in their mouth if this fish tastes nice. I don't even know how it tastes. But out of total, 42% of these samples also had these resistant bacteria. And again, this is one of the screenshot taken from aquaculture, also from the ICMR report. So just this is the last slide from my side for epidemiology. I'm really sorry I was a little quick and little faster in this time, because we have to finish this workshop on time as well, and we should always respect time. So I quickly wind it up so that I can hand over the stage to our next speaker, who is from EFSA. But before I invite him, I would like to give a brief background. All these scenarios which I've just shared, this is the percentage, this is the percentage. So is there any impact? Is there any negative impact? One, for sure, is our health. Our health is being impacted when we are eating it, right? And it's as we have already seen, it is not only related to non-vegetarians, even vegetarians, because we are having milk and other dairy products. So everybody is being impacted. So one is health for sure, but secondly, it is also impacting our economy and how our economy is being impacted. In 2016, the European Union took one decision, and they rejected consignments from India. As sir has already showed, Dr. Himanshu, that India is making a lot of production from meat side, whether it is cattle, whether it is aqua, but we got rejections on the export of these shrimps. And why that happened? Because which should not be there in these food materials. So this is really alarming. This is impacting our economy as well. And as I've already told, I've shown you in the beginning, intentionally I had put in that data compared to the European countries, that they have very less amount of antibiotics in their system. So what is that they are following and we are not following? So in this regard, I would like to request, I would like to end my session here, and I would like to introduce our next speaker, Dr. Ernesto Libana, who is a veterinary surgeon with a PhD in microbiology, and he joined one of the reputed organizations. I am sure all of us must have heard this name, alike to WHO, at some point of time in our lives. That is European Food Safety Authority in the year 2007. And he's the head of biological hazards and contaminants unit, which specifically looks into these kind of situations. And he also deals directly in the departments of food and food and environmental safety and pathology in the veterinary laboratories agency in UK in his earlier times. And during his research, his prime focus was also, in addition to other aspects, his prime focus was also on the development of antimicrobial resistance in these organisms, how antimicrobial resistance is happening. And now he's also actively involved in food scenario or food aspect of this situation. So again, as I've told, I can keep on speaking in his from his CV, but I have to respect the time, and I would like to thank you, Dr. Ernesto, for agreeing to our request. And over to you. And he will be discussing basically on the European perspective on monitoring and risk assessment of antimicrobial resistance in the food chain from European perspective. Thank you so much. Over to you, Dr. Ernesto.
Thank you very much. Good morning, good afternoon, everybody, and thank you again for the invitation and the chance to be here with you today and try to explain a bit our experiences in Europe. I think you made an excellent start by, sorry, I'm just trying to put my slides on show mode. I hope you can see them. Yeah, okay.
Still, we are unable to see. I was about to request you for that.
Okay, sorry, because for some reason this is not working. I'll try again.
All right, no problem. We can wait to listen to such expert, not a problem at all.
Okay. It should be working, but for some reason it's not.
Maybe you can try once, otherwise it's okay. We can see the slides. Yeah, it's perfectly visible.
It's
great.
Let me one. So I was saying that, yes, I'll try to explain with this very nice background that you and your colleague have provided. I'll try to explain how do we tackle this problem in Europe and what we have done in the last kind of 15 years. Just the first thing, I will not be talking that much about the problems of residues in food, just because for us, after implementation of a very strict policy in the marketing authorizations for veterinary medicines and for using any antimicrobial in treating animals, you need to have a marketing authorization. And then there are very strict rules also on these withdrawal periods that were mentioned before, meaning that when you treat an animal, you have to wait for a certain number of days before any food can be consumed by a human. And we also have a very strict residue monitoring program. So all these things together have resulted in very low levels of residues. So we can safely say that the food that we consume in Europe does not have antibiotics in. So that is something that at least we are confident about. But there is the second problem that was also presented, that is the presence of resistant bacteria or resistance determinants, meaning antibiotic resistance genes that could still be on foods. And that is the problem I will be tackling mostly in the presentation. So to start with, one thing which is really important is to have data. As you have seen in the previous presentation, without knowing the level of the problem in your own particular environment, it's very difficult to do anything. So it's very important to collect data on resistance, and we do so for all the countries in the European Union. But it's also important to know the volumes of antimicrobials that are used to treat our animals. And then when we use this data and we combine this data together, we end up with a powerful, convincing mechanism to tell governments that they should start doing something about reducing the problem. Because we are showing also the links between the levels of resistance in animals and the levels of resistance in human pathogens, so in the bacteria that give humans disease that cannot be treated. So I have to emphasize that the first step that one should take is really to gather data to understand where you are in a scale of things with this problem. And then how can you be successful in getting data? And the European experience has also been that you can only get good data if you have law, if you have legislation that drives, that makes compulsory for, in this case, the different countries in Europe to collect this data. And this data has to be collected in a way that is harmonized and comparable. So you want everybody participating in the monitoring and surveillance schemes to do things in the same way. So this can only be done really if you have clear law behind. So for a number of years, we have been working on defining these laws and improving the way the monitoring has been done. And the most recent piece of legislation is here, this Commission implementing decision from only last year. And in this piece of legislation, we specify everything. We specify the animal species that have to be monitored, the bacteria, the antimicrobials, the sampling schemes. Everything is very clearly that the laboratory methods, everything is completely clear so that the member states know exactly what to do. And then there are important components in any monitoring scheme. So you have to define the target population. So which of the animals, meat producing or milk producing or egg producing animals you want to target, depending on how important the production is in your country. You have to decide which are the bacteria that you are going to monitor. So in our case, you see here that for the different animal populations, we have selected different bacteria, things like Campylobacter, Escherichia coli, Salmonella are our main target organisms that are compulsory in our monitoring schemes. You have to define also the number of samples and the number of isolates per year that you want to investigate, because this number has to be representative of the animal population in your country in order to allow you to draw comparisons between countries. You have to define where you take your samples from, so the sampling point. And in Europe, this is mostly done at the slaughterhouse level, although in certain cases, we also do the sampling directly in the farms in primary production. And this is due to the fact that we also have schemes for controlling certain foodborne pathogens such as Salmonella. So we want to maximize the usefulness of the sampling. Basically, you wouldn't have to sample for Salmonella at the farm level and at the abattoir level. If you are already going to the farm, we can take the samples there and use them also for the antimicrobial monitoring programs. But it's important that you define all of these. And very important that you are clear about your sampling design. So this is what I talked about, the representativeness of the sample. And this has to be stratified to the national domestic production and the animal populations, which are more prevalent in different countries. So this is a kind of homework you have to do first to design your monitoring program. Once that is ready, The other thing that is very important is that you should be transparent with the data you collect, meaning that in the case of Europe, every year there is a report that summarizes all this information, all the results that we get from the monitoring. And more so, this is a One Health report. So it's a joint report between us in EFSA that we deal with food and food producing animals, and our colleagues in the European Center for Disease Control that gather all the data on resistance for human pathogens. So the data is presented in both populations in the same document. And we cover because this report is mostly done for public health purposes. So we cover all critically important antimicrobials for humans. We calculate also temporal trends, because as we do things in the same way, year after year, you can compare your results and see whether the situation is improving or not. We also have, because we monitor so many animal species, so many antimicrobials, so many bacteria, we want to have also a number of succinct, what we call key indicators that we use to draw these temporal trends and understand whether the management measures which are in place are having an impact or not. And I will explain which ones are these. And we also keep an eye on what we call emerging risks. So from time to time, there are new resistance mechanisms or resistance to important antimicrobials popping up in animal bacteria. And we want also to give in these reports this angle of telling the readers what do we think is more emerging or more dangerous for humans. And I don't want you to really concentrate too much on the graph here, but what I want to give you an example is how do we present the data. So you have, for the different animal species, you have the percentage of bacteria that we collect which are resistant to a number of substances. And normally what we observed in the case of Salmonella and E. coli is that for those antimicrobials such as ampicillin, sulfonamide, or tetracycline, which are very commonly used to treat animals, we see from moderate to extremely high levels of resistance. Luckily for us, we still see low levels or very low levels of resistance to important antimicrobials for humans, such as third and fourth generation cephalosporins. And we have a very low level of resistance when we combine resistance to fluoroquinolones and, in this case, cephalosporins. So the situation is not perfect, but at least we have seen it is improving in many cases. And we show, as I mentioned, data for all member states. And this is very important because then countries see the results also for their neighbors, and they can compare to each other, and nobody wants to appear in a bad light here. So those countries that have higher proportion of resistance perhaps need to think about what to do or how to learn from other countries that perhaps have much lower levels of resistance. So this is a very important motivator where countries see their data together, it fosters action normally. And in this graph in particular, I'm showing the prevalence of ESBL and AMSI producing E. coli. So these are bacteria which are resistant to an important group of antimicrobials that are used to treat critical infections in humans. And we have seen that the trend, although you still see countries such as Spain or Italy with high proportion of resistant E. coli, the trend is overall decreasing. So we have seen that in 48% of the member states, we are observing a decreasing trend in the last few years. We have seen also that we converted, in this case, this ESBL AMSI producing E. coli into a key outcome indicator. And you see all the countries here. So you see that the different countries have very different situations. And overall, as I mentioned, we have a decreasing trend in 13 member states, which is, I think, positive news. Another key outcome indicator that we use, and it's very important for us, is what we call the complete susceptibility. So this is the percentage of E. coli that are fully sensitive to all the antibiotics that we have in our testing panel. So the higher this indicator is, the better the country is performing in tackling the issue of resistance. And as you can see here, we have marked variations of this indicator, and there is a gradient. So normally, the higher up north you go, the better the situation is. And the same thing tends to happen from east to west of Europe. But again, this information is all fully publicly available and transparently reported. And this shows, as in the previous indicator, that the situation is very different in different countries. And overall, we are seeing these increasing trends in fully sensitive E. coli in 25% of the member states. So this means that what we are doing is somehow paying off. We are seeing the first results after all these years of efforts. Okay, so this, I presented a bit what we do for monitoring data on resistance, but I said that it's also very important to gather data on the volumes of use of the antimicrobials, and also to do a correlation analysis between the levels of use and the levels of resistance. And we do this together with another two sister agencies in the EU, which are the European Centers for Disease Control and the European Medicines Agency. And we put together all the data from animals and humans on use and on resistance. And we do all sorts of correlation analysis. So we correlate we do an analysis of the amount of antimicrobials consumed in animals and the resistance levels found in animals. Equally, the amounts consumed in humans and the levels of resistance in humans. And then we do also across comparisons to try to understand what is driving finally the resistance. So we have done this for we have done three of these reports. We produce them every 2 or 3 years. And for the first time in the report this year, we have seen that the overall consumption of antimicrobials is lower in animals than it is in humans for the period 2016-2018. So again, this is showing that the veterinary sector is really taking the problem seriously and putting into place measures to decrease, you can see this green line, decrease the amount of antimicrobials that we are using to treat animals. While in humans, the situation has not really changed dramatically. So sometimes you are seeing a slight increase, but certainly not a decrease. So perhaps that gives you an idea also on which sector you should be putting more efforts right now. And by doing these correlations analysis that I mentioned, we have proven, for example, that in the case of humans, there is a clear, significant, statistically significant correlation between how much carbapenems, the levels of carbapenems that you use to treat humans and the levels of resistance that you observed in humans. And you can do this for many of the other antimicrobials. And for example, in Campylobacter jejuni, it's clear that the amount of fluoroquinolones that you use to treat animals are correlated with the amount of resistance that you find in those animals. And in turn, those levels are correlated to the levels of resistant Campylobacter jejuni infections in humans. So now suddenly with this analysis, you can tell your risk managers, your politicians, hey, you have to do something about this, because now we have given you proof that the more antimicrobials we use in our animals, the more resistant infections we are seeing in humans. So they have the hard scientific evidence they need to put action in place. So the conclusions from this type of work is that reducing the consumption will have a beneficial impact on reducing the resistance. It seems like an obvious conclusion, but believe me, unless you give the real hard data to your politicians, they will not do anything. So it's the first thing you have to do is to prove this. And therefore, it is important to promote prudent use of antimicrobials at all levels in animals and in humans. to have sufficient infection control policies and prevention of infections. And these interventions really should be reinforced in all countries. So this is what we do now on correlating the different data streams, but there is another part of resistance that we have not really explored in Europe, and this is the resistance in those bacteria that only cause infections in animals. So until now, I have been talking about E. coli, Salmonella, Campylobacter. All of these are zoonotic microorganisms. So they are bacteria that you find in humans that can be transmitted to, in animals that can be transmitted to humans. But it is also interesting to know what is the risk of developing resistance in those bacteria that give animals disease, and whether this is also compromising the way we treat animal infections. So we are in the early stages of this work. So we are working in what we call a mandate, so the set of questions that the European Commission sent to EFSA, with the idea to end up with a list of the most relevant veterinary pathogens that show a higher risk for development of resistance, and whether we should also start building up a monitoring program similar to the one you have seen for zoonotic pathogens, whether we should start also monitoring this in animals. So this will come towards the end of this year. We will already be seeing some documents published in the EFSA website. Now, everything I talked about was only showing data on what we call phenotypic resistance. But until now, there has not been any effort to do a systematic and harmonized surveillance on the genes which are behind the resistance levels that we observed. But nowadays, with the mainstream application of genomic technologies such as whole genome sequencing, we can now do also this. And we realize this is challenging for countries because they are at different levels of technical ability, and not all of them are able to jump on board and do this. So the decision has been to do an incremental introduction of these newer technologies over the next few years. And we started already developing harmonized protocols, training the different countries, giving them the bioinformatic tools that they need to do, helping them with their sequencing platforms, etc., etc. So with time, we will be able to have also in our reports data on the molecular level, on the genes which are behind. And this is very important because it will give us also the possibility to do more in-depth epidemiological investigations. So really to give us more insights on the transmission directions, whether we are seeing the same bacterial clones and the same genes moving from animals across to humans, or whether it is vice versa, that sometimes you get permeation of bacteria and genes coming from the humans and contaminating the animal production environments and amplifying the magnitude of the problem. So all of this is coming our way. It's a very exciting time to also introduce this type of data. But then once you have the data, you have to use the data. And for that, we do what we call risk assessments. So we use all this monitoring data, and over years, we have done work on methicillin-resistant Staph aureus, on ESBLs, on carbapenemases, and we have given the risk managers, those that need to take decisions on what to do, enough scientific evidence to put in place certain measures. And one of the most important pieces of work we did is this document that we call RONAFA, which is providing advice on how to reduce the levels of antimicrobials use and how to provide also recommendations on alternative to antimicrobials. What we have learned over time is that it's very difficult to conduct these farm-to-fork fully quantitative risk assessments for a number of reasons that you can read on the slides, because the hazards are not only the bacteria, but also the genes, because there are many different possible routes of transmission or acquisition by humans. You have the food, the direct contact with the animals, the contact with the environment, the transmission from human to human. So it's very difficult to model all this complexity. On top of that, you have the issue of imports of foods and also of international travel. You don't really have good data on the numbers of bacteria in the foods. You know the proportion of isolates which are resistant, but you don't know how many bacteria in the end, resistant bacteria you have in the piece of food that you consume. You have very diverse consumption habits in different countries and in different types of consumers. In general, those response models are very complex, and all of this makes quantitative risk assessment quite complex. So coming back to this advice we gave, this RONAFA opinion, and I invite you to go to this link and read a bit the document. We ended up with recommendations in 3 areas. We call the areas reduce, replace, and rethink. So in order to reduce the volume of antimicrobials, there were measures, important measures such as setting targets at sector level or at country level, so that you could allow benchmarking. This is the same principle I mentioned before. When people compare their farms or when sectors compare across each other, they see that you have good farms and bad farms, and they don't like that. So it is pushing people to act if you set these targets. So you have to say by year, I don't know, 2025, I want a reduction in the volume of use of whatever, 5% or 30% or 50%, depending on how stringent you want to be. Then it is very important to enforce that antimicrobials can only be used under prescription. So you need to have veterinarians prescribing, and farmers do not have direct access to medicines without medical advice. And very important, phase out all preventative use of antimicrobials. So antimicrobials should be used to treat infections, but not to prevent infections in animals, because we have seen that in the past, the levels used for prophylaxis were far too high, and this is a problem. Then on the replace front, we analyzed the number of alternatives, the use of organic acids, probiotics, competitive exclusion, bacteriophage. Many of these substances, the problem with them is that they were used with an outcome more geared towards production and not health. And sometimes it's also difficult for companies to embark on producing these alternatives because there is not a legal framework clear for them to market the products. So we gave advice on what to do to improve this situation. And then on the third part, which is the rethinking, it is very important to have proper farm health plans and treatment guidelines for different diseases, fostering the use of vaccines, and also doing a critical review of these intensive systems that are high users of antimicrobials. So in other words, looking at what can be changed in husbandry conditions to make animals healthier and happier, so that you need to use less antimicrobials to treat disease. And it is important to remember that there is not one single measure that is a magic bullet. You have to implement all of them. You have to think about all 3 areas in order to be successful. And that was a very clear message. I'll move on quickly because I know I'm taking a lot of time. Also, very recently, we looked at the importance of the food production environment. So not only the animals, but also their environment in the problem of resistance. And we were able, in a document that was published last month, we were able to talk about sources and transmission routes, and the importance of fecal matter contamination used in as fertilizing agents, and also in the contamination of irrigation water. We also looked at what bacteria and genes of public health priority were present in these food production environments, and a lot of them, like ESBLs, carbapenemases, MRSA, QNR, all these genes and all these important bacteria are present in the food production environment. So very important to look at general factors such as biosecurity, hygiene, good farming practices, There are certain things, for example, replacement of animals and persistent environmental contamination that needs to be looked after. And also we have to remember that in general, the microbiota of natural environments are a source for genes. And this is a very difficult to control source, of course, because they will always be present there. We talked about different mitigation strategies here, like reducing the bacterial content of manure, sludge, and irrigation water, and also preventing transmission from other animals by implementing good biosecurity. And also in the case of aquaculture, for example, ensure that there is high quality of the water by reducing and eliminating contamination of the water with fecal waste. And finally, last but not least, we identified also knowledge gaps and provided a number of recommendations to the European Commission so that they should put money in certain areas for research investments to be able to generate more data. We are also now working on a particular mandate, which is looking into residues. I started my presentation saying that residues are not really a problem in the EU, but because we are so stringent, and many years ago, we already discontinued the use of antimicrobials for growth promotion, we want to make sure that we don't go back to past situations. And I'm sure you may be aware that the way we treat our animals is very different from the way we treat humans. And in many cases, you have to administer antimicrobials via drinking water or via medicated feed. We have a problem in which the normal feed that does not contain antimicrobial and the medicated feed are produced sometimes in the same facilities, in the same feed mill. So you may have the risk of cross-contamination from the medicated feed to the non-medicated feed. And that is why the European Commission wanted to set what we call maximum residue levels in these feeds, so that this would not pose a risk. And they gave us a very challenging mandate for 25 substances to come up with these levels, these maximum residue levels in feed that would not be a risk for selecting resistance or for having a growth promotion effect. And just to cut a long story short, what we found is that very low levels, and certainly well below the minimal inhibitory concentration, the MIC level, very low levels of antimicrobials do already promote selection and spread of resistance. So we came up with these values, and now the European Commission will have to decide how to legislate about that, how to set these maximum residue limits. And this takes me to the end of the presentation. So just to summarize, how important it is to have comprehensive, harmonized data with representative sampling schemes for all these species and compartments concerned. The importance, increasing importance of the new molecular approaches. Also, the conducting these associations between use and resistance. This is very important. We have many examples from member states where they were stringent in setting targets and reducing the volumes of use, and they have seen that this pays off, for example, in the Netherlands, with a very large reduction also of the percentage of resistant microorganisms. And I also explained why the fully quantitative microbial risk assessments are difficult and sometimes with high uncertainty. So we cannot wait also to have this completed. We need to act before. And if you are interested in all these documents, you can find them in our EFSA webpage, or if you have specific questions, you can also contact me, and I will be very happy to provide help with anything I can. So thank you very much.
Thank you very much, Dr. Nestor. It was a very nice talk, very informative talk, and I am sure it was an eye-opener for most of us. Now we know at least in which direction we should move. But as you said, we should transfer this information to our higher authorities so that they can take it forward in a better way, because they are the experts in this field. All right. So before we move forward, Dr. Nestor, I would request you to stay back here only. And I would like to thank all the students as we're running a little late, but we'll surely try to, wind up little early. And I'm really sorry, dear students, we are skipping the break right now. And I want a little bit of your patience. And I promise that we will try to be, more on time in our next workshops. Anyways, I'm not taking all the questions, but one question to Dr. Himanshu and Dr. Ernesto. So I would like to start with Dr. Himanshu. Before that, yes, thank you to all the speakers as well, because you are also taking time from your busy schedule. We will soon start with the debate section. All right. So, Dr. Himanshu, there is one question for you. The student has asked that antibiotic resistance contamination can occur in production, processing, and as well as handling of animal food. So, can you elaborate a little bit on that?
Yeah. Okay. Thanks, Dr. Kiranjit. See, I mean, there are different ways that antibiotic resistant contamination happens. Suppose, for example, you are producing manually, you are producing animal feed. Suppose you are giving, food to the chicken, or you are giving food to the cattle. Suppose, first things you are mixing all sorts of ingredient during. Suppose that ingredient getting contaminated with contaminated water, which has already antibiotic residue or antibiotics, or it may come contact with the soils, or it's the preparator who prepare this thing. If you are not properly hygienically maintained, he or she may carry also, resistant bacteria. So during the process, it may come. This is while production, and during processing also same thing, because when you are mixing together manually, even if in machine, even if industry, if the hygienicity is not maintained, during process also get contaminated with the water and ingredients. So during the process, either production or processing, also food also getting contaminated.
Yeah, thank you so much, sir. All right. So I move to next question. I hope Dr. Ernesto will find this question very interesting. So we've got a question that, do you think initiative like RONAFA, which you just mentioned during the second half of your presentation, can be implemented easily in Indian scenario with such high population and such high number of food animals? So do you think that can be directly implemented? I think this is what student is trying to ask.
Yes, I think it's not really dependent on the size of the population, or in this case, the size of the production, because as I mentioned, there is an array of possible different measures, and I think all of them are implementable to some degree. And I think it is important to understand that. I think the main message from that piece of work is, as I said, that there is no single measure. So you cannot expect that by doing one thing, you will see a difference. You have to do many different things, and the combination of those then will have an impact. So that is the important thing, to concentrate on putting a little grain of sand in all the places where you can, and then you can build a mountain. Yeah, thank you.
Great. Thank you so much, Dr. Ernesto. All right, so I would still request Dr. Ernesto and Dr. Himanshu to please stay back in case there are any other questions we have for you from the students. All right, so moving forward toward the last section of our workshop, that is the debate section. So I would like to request Dr. Alfonso, Dr. Prakash, and Mr. Joydeep to kindly keep your cameras on, and keep yourself unmute. And this also gives me a good privilege. I think we are missing Mr. Joydeep. Maybe I would like to request, I'll just try calling him. Maybe by that time I can just introduce the other speakers. All right. So Dr. Alfonso Zacconi, he is a professor of epidemiology and infectious diseases, University of Milano, and a former director of the specialization school in milk and milk products, hygiene and technology. He is an expert in IDF, SC, animal health and welfare. His research topics cover epidemiological studies on risk factors for mastitis, about which Dr. Himanshu was discussing, development of an immunological model to assess immune status of the cows at blood and milk level, development of dairy herd health management programs, so on and so forth. So the moment you think about anything related to animal health, I think specifically milk products, Dr. Alfonso has experience in that direction. So we are overwhelmed to have you, Dr. Alfonso. Thank you so much for joining.
Thank you.
Yeah. All right. So then we have Dr. Prakash with us. He is currently working as an associate professor at Prasanna School of Public Health, Manipal. He is actively involved in teaching and research, and his area of research includes, again, as I've told, that we have experts who are working in multiple fields as well. So we have interdisciplinary situation here. So sir is working in multiple domains of both communicable and non-communicable diseases, in reproductive health, nutrition, digital health, zoonosis, drug resistance, and he has many papers to his contribution. And he's also guiding various students at postdoctorate and master's level. I think this holds true for all the experts which we had. So I just try to pick and choose, few lines from different expert series. All right. So I think we still miss Mr. Joydeep. We can wait for him in case he's able to join. But just for the audience, I would like to mention that Mr. Joydeep is a coordinator from AVAT, which is an NGO. The key idea is that till the time this information doesn't go to individual, maybe the farmers or the milkman, then this information is of no use. As I mentioned in the beginning, the more we share, the less it is, because this is very important information, and food is part of our system, and we cannot survive without food. So at least the producers, especially farmers, milkmen, they should know about all these details. All right. So we move towards this section, and as the name indicates, this section is a debate on food safety and AMR, break the chain. So till now we have heard a lot about problems. This is the problem, this is the percentage, blah, blah, blah. And I think most of the students must be sad also that what we are eating, is it healthy, not healthy, but please do not worry. The good thing is that we are having this workshop, we are discussing this problem, and till the time we don't discuss, we cannot come up with a solution, right? So let's listen from the experts. And in case you also have any question, you can also put in the chat box, and I can also use that question for the debate section. All right, so my first question is to Dr. Alfonso. Dr. Alfonso, as discussed previously by the speakers, antibiotics are being misused in animals, that is for sure. So since you are associated with animal health and welfare associations directly, so what actions do you think are taken specifically to break this chain?
Yeah, thank you, first of all, for the invitation to this webinar. It's very interesting and really learn a lot on listen to the different speakers. So I'm really thank you for that. I'll try to answer to your question, which is not really easy, but in some way, Dr. Libana helped me because, as you know, he said, at least in our experience in Europe and Italy in particular, legislation was implemented since 15 years ago. So we start to have a ban on growth promoter in 2007. So and year by year we have new regulations. So this is what I call a carrot and stick approach. So this is the stick. So the regulations make stricter the use of antibiotics, and we are not allowed now to use some of the molecules in human in veterinary field because they are reserved to human. And starting next year, we will the use of antibiotics as a preventive measure will be banned in Europe. So in the field of dairy cows, a very common a way to control this mastitis, for instance, is to use dry cow therapy, to apply antibiotics at drying off. So this procedure will be banned starting January. So this is the stick. Regulation that are in some way give the results. But as Dr. Libana say, we have no residues or very few cases of residue in milk. And this also was to give a broad aspect. This was also obtained by fines. So most of the European milk is processed by dairies, and dairies fine the farmer if they found antibiotics in milk, and the fine is very high. So this again is a very hard stick. So when they're fined, they're not delivering milk with antibiotics to the dairy. The kind of side is to increase the awareness of this aspect to the people, to the whole chain, starting from the farmers to the veterinarians and to the technicians. So in this way, you have to make clear the problem, and also to try to, as we are doing now in this webinar, try to give some solutions. So the importance is to give them a way or system or protocols to improve the health and the welfare of the animals. Obviously, we can not go to specific issue in this webinar, but obviously there are a number of procedures we can apply to reduce the risk of disease. Reducing the risk of disease, we are increasing the welfare of the animals and to decrease the risk of treatments. And this is working. For instance, in the next 2 months, I will to travel around my region to explain to farmers that they cannot use the antibiotics as a preventive, but then have to implement other things to avoid to use them. So that makes as we are doing this webinar, to spread the knowledge, to give information, to give advice, and to try to improve that. This is a part of the, could be progressive, you need step by step, but this is very important to obtain the results, because the stick is not enough. And then you have to, and this could be applied for other things, excuse me, on intensive farm or to family farms. The principles are the same, so you can work on that.
Great. Thank you so much, Dr. Alfonso. That was a really nice explanation, what you could have given to us on this question. And rightly said, there is, the more we create awareness, the more is the, good impact of this scenario. All right. There are two announcements which I would like to make before I ask the next question to Dr. Prakash. One is Mr. Joydeep has joined us. Thank you so much, sir, for joining. I think he's facing some internet issues, so we hope you will stay back, during this session. And second, Dr. Ernesto and Dr. Himanshu, in case you feel that you can add on to some information during this debate section, you are more than welcome to add the points. All right. So moving towards you, Dr. Prakash, I would like to request to unmute yourself. So in continuation of the question which was asked to Dr. Alfonso, he mentioned about, dry cow therapy, stick and carrot thing. So there are many guidelines and, the main guidelines for avoiding the use of antibiotics in animals that has been implemented since ages. Maybe you can say since ages, because we are in 2021 and compared to 2007, it's like a long time. So what is the scenario of India in this context? Do we have regulations, in place, or is it that where are we missing?
So, am I audible?
Yes, you are.
Thank you. Yeah. So, thank the organizers and the eminent speakers on educating us on this important topic. Yes, and this question on where are we at this point of time, timely, and yes, we have guidelines on tackling antibiotic use and antibiotic resistance in India, and yes, but again, rules and regulations are there, and implementation needs to be enforced, and that's the major issue. But okay, but different sectors I can mention, like one is this food safety and the standards association. So they have issued revised guidelines from the 2000, early 2000 onwards. They started issuing guidelines and advisories on how to address this issue. So they started having sort of guidelines, and they try to create awareness at different stages, like from the production, processing, and even the cooking practice and so distribution networks and cooking practices from farm to fork. So that policies are there, and then this national antimicrobial resistance policy, national action plan on antimicrobial resistance, that came fairly late in India, maybe 2017. And again, the guidelines been set by the central government in consultation with the WHO and other partners. But the implementation, again, it's different in different states. So the central government called for a meeting, and the state government participated, and they've been asked to frame appropriate interventions for their state. So that is in process, and certain states are really good at implementing some of the guidelines, and you can see some of the, I think you mentioned some studies, Dr. Siranjit mentioned some of the studies. So many states started implementing and started monitoring the resistance and use of antibiotics in agriculture sector, animal sector, and human sector. And some states are yet to implement it. And that part is because of COVID and plus initial target was from 2017 to 2021, we are supposed to achieve some reduction in the antibiotic use and resistance also. So budgetary allocation for last 2 years is very poor on this particular aspect. So that is one of the sad part. But and plus this pharmaceutical companies, or you can say regulating pharmaceutical drug use. We also have regulations on, and they from time to time, say from 2007, 2012. So we have identified certain group of antibiotics, called Schedule H. Initially, some 500 plus drugs being identified, and that should be dispensed only at the prescription of providers. And then again, they expanded the list recently, and then another set of guidelines came recently, that is Schedule H1, that is compulsory, and then other further restrictions on prescribing and use of certain antibiotics. So things are moving in a positive direction. And cosmetic acts are also talking about use of animal products also, and plus milk, honey, animal products as well as meat. And they are this safety, they are talking about reducing the antibiotics before they start selling or using the product for human consumption. So they're saying they put particular dairy products and the chicken, at least you stop providing antibiotics at least 1 week before you start processing the particular meat or products for human consumption. So different rules and regulations are there in place, and plus we set up a chain of laboratories in human and animal sector to test and kind of surveillance, we can say. So we are looking at the model of which is there in Europe, and we have India, the state constitutes like a mini Europe. There is a lot of constituent states, so then different political systems, and so so that's one of the challenge. And yes, so animal sector is one of the better equipped to detect most of the this resistant pathogens as compared to the human sector. But so with this new policy, they set up around 10 surveillance centers already in place under Indian Council of Medical Research, and they are trying to set up 20 medical colleges for, I think, 20 or 25 medical colleges for the surveillance purpose. So I think overall the scenario is improving, but there are a lot of things to move. That's what I could say at this point of time. And
it's good to hear that we are working in multiple directions, like you mentioned, in pharmaceutical, in drugs and cosmetics. So it's good to hear that it is also happening at state levels. And for sure, yes, COVID has a negative impact on this, on all the aspects. So why this would be taken aback.
But it was, it was supposed to be an opportunity. Now it's disease, and it's again zoonotic. There are discourse on is it a zoonotic or not? It's SARS-CoV, but it can be one of the assumptions. So we have to work in maybe collaboration, multi-sector collaboration is very important, and this was one of the time to improve. Maybe it's happening at different levels. Okay, thank you.
Yeah, thank you, sir. All right. Now moving again back to Dr. Alfonso, as we have heard during his, brief CV that he is expert of milk products and dairy, area. So I would like to know that, like you mentioned, that in next, I think maybe I forgot the timeline which you mentioned that you're going to visit the farmers, for creating awareness and all. So, what do you think that a producer, a milkman, can do to ensure that, or to break this chain, that how this antibiotics are being spread? So, I think you are the best expert to have, give a suggestion on this.
I think that to do this, you need to apply very simple things. First of all, one thing is common to farmers all over the world, so it's not a problem of size or of location, is that the farmers should know that antibiotics are not a solution. It's a tool to change, obviously in a positive way, the health of the animals. So this will be, if you want to keep the animal healthy, you need to work in a proper way. That means you have to feed them properly, obviously based on the yield, the breed, but also the environmental conditions. So I expect that what you can do in India will be a little bit different from what we can do in Europe, for instance. But
Yes.
the principle should be the same, to keep the animals at our best welfare and health. And this you need to have to work in a hygienic way. So for instance, one very key issue is at milking time. So if you milk by hands or you milk by machine, hygiene should be there. So obviously you can wash the machine, the hands, or wash the machine if you want, but then the principle is the same. So when you apply very simple things and simple principle, hygiene, good nutrition, in general, good welfare for the cow, think of the cow, not think of you in relation to the cow, then you reduce the number of diseases, including obviously mastitis, that
Yes.
is known as one of the major sources of use of antibiotics. And then you can improve the situation. So in some way, think simple and keep the thing simple. That will help a lot. The other thing is to not to think that alternatives or magic bullets or magic powder could do the work of a proper diagnosis, a quick intervention on the animals. So it's important to be very rational from this point of view. So and this again will help to reduce the risk of antibiotics in milk on the environment, and to improve anyway the quality of the milk. So improving also the nutritional value of the milk.
Rightly said. So you have touched upon the root of the problem. So if animals are healthy, then of course we do not need antibiotics at all, right?
Yeah.
And then they will not enter into the system. Then we also don't have the situations of withdrawal and other things. So if you work on the root, then you don't have the other one.
Yeah.
Very rightly said.
Yeah.
Yeah. So I will continue with you only, Dr. Alfonso. Since you are the expert of milk, so I am sorry, but I'm also going to ask you another question related to the same. There was one of the report wherein I emphasized during my talk. I'm sure you must have taken back your attention. In one of the studies, I think that was probably from Karnataka, wherein they have found out that if they heat the milk, the impact of antibiotics reduces. So, what is your call on that? Because normally it is being told that if the milk is pasteurized, it is like pure like anything. So, what is your call on that?
So you already emphasized that you can reduce the risk of antimicrobials or others through heat treatment. So again, put in a simple way, the higher is the temperature or the time you apply to the milk, you decrease risk for bacteria or risk for antibiotics or residues. We are decreasing, you are not going to zero. And but on the other side, the higher is the treatment in terms of thermical treatment or time or application of the heat, you are decreasing dramatically the value of the milk as a nutritional factor. So as you know, the heat treatment is based to reduce the bacteria, the pathogens. So, and this is important. The pasteurized milk could be a good example. You have bacteria in pasteurized milk, you don't have pathogens. So if you have bacteria, you can have also bacteria delivering genes for resistance. So, but when you have pasteurized milk, you have a nice food for human being. So again, if we keep this milk as healthy as possible, we don't need to treat with high temperature. And then you have a much better food for human being. The more you manipulate, you can say, the milk, the less is good for human being. And maybe have less risk, but it's also a much lesser value from nutritional point of view. So I will stay with I don't say the raw milk is because it could be really dangerous, but pasteurized milk should be a benchmark to work on.
Yeah, great. Rightly said, Dr. Alfonso. Thank you so much. All right. So since we are towards the end of this workshop, I would like to quickly ask another question to Prakash sir. So since now we have listened to Dr. Alfonso from the producer point of view, that what a milkman should keep in mind while producing the milk. So anything which you would like to emphasize on what a consumer should keep in mind while taking these products? So you need to unmute yourself. I'm sorry, and I would also request you to just take a couple of minutes. So that
I think this concept already discussed in by previous speakers. So at every step, like Professor Sargoni has clearly mentioned, awareness creation of the producers, whether it's the meat or what is agricultural product. So that's the very basic step, and that So and plus other aspects, but at every level we have to ensure that they are not misusing antibiotics for maybe growth promotion or other. And plus, I just want to add a bit on the other environment aspects. Like say the land, or you can say soil pollution and air pollution, water pollution. That also will get into this one. And plus, in India, what we find is certain places this grazing land where these animals are grazed, maybe in the forest fringes. So there it's a chance like Professor Libana mentioned, the infections from wild animals can get into the human or even our domestic animals. That is one of the risks, and we really don't know which of the bacteria or viruses resistant in other animal sources. So that is one of the major risks, that can, the animals will get those resistant strains, and, that will pass on to the people who are going to consume it. That's one of the, then which you can, many places you can see individual household farms or even small farms, maybe in the forest fringes. So that is one of the major thing I want to add. All of this other points mostly mentioned, and the surveillance of all these products, food products, time to time is very important. But again, I want to emphasize that. So thank you very much.
Thank you, sir. All right. So this brings us to the end of this workshop and this debate section. Only one last question, maybe to all the speakers. So anyone who is interested can unmute and answer. Although we have discussed and we have listened to your expert experience already, that what all steps we can do. But specifically in if we talk about a developing country like India, so any specific suggestion from your end, which can be one of the steps which India must emphasize and must take forward so that we can break this chain. So open to the floor. If anyone would like to give any final comment on this or would like to suggest something.
Can I add a point here? One is we have scheduled health drugs for human use.
Yes.
The veterinary sector also have certain list of drugs, but the enforcement is one of the aspects which I think India is lacking at this point of time. That I would like to add, and I'm eager to listen to that. Thank you.
Yeah, great. I think Dr. Alfonso, like he also mentioned about the fine thing. I think if that stick thing will be here, so I think that is going to, bring a lot of change. Like same happens in our class. If we don't have any, repercussions mentioned, then the students also don't study. So, okay. Still, if anybody would like to add some point, you're more than welcome.
Yeah,
okay,
perhaps,
I see.
Perhaps
please,
can you
Dr.
just
Ernesto.
let me, I think for me, what is crucial is get your data as soon as you can. And then I
Great.
think what you've done today is really brilliant, because is once you get the data, and in your presentation, you were presenting some specific data for India on certain species, on certain foods. Make those data as publicly accessible as possible, explain it to students, explain it to consumers, and then this will turn into some pressure for the politicians to do something with it. So I think that's really the beginning of everything. If people are not aware of the problem, very little.
Exactly.
So that's for me the most important.
Yeah, thank you so much, Dr. Ernesto. All right, I also see Dr. Himanshu. Yes, sir.
I think last point I can give, because the most important thing, awareness level. That is the genesis of this preventive project. Awareness level of antibiotic resistance. It is starting from educated people, illiterate people. That awareness level needs to be enhanced. Because even though we have policy, even though we have programs, because until unless this concept percolate down to the end user, the common man, because then only we can do a change. This is one aspect. And second aspect, also this all the speakers, they have highlighted that all this policy protocol, whatever there, that needs to be right spirit and right way, it needs to be implemented. And third thing also the data. We have to develop evidence, because so that it will facilitate for stringent policy development. So these are the three things we can look forward. Thank you.
Perfect. Thank you so much, Dr.
If you may add the last thing.
Please, sir. Please.
Based on also to Dr. Lebana's statement, I agree that data are important, but data should be also used to define objectives and to have a roadmap to go there. So this is important, not to be led by emotions or by only pressure from public opinion, but sometimes the pressure are not correct. So when you have data, you can build up a roadmap and to stick on objectives. This is important to come to the target.
Exactly. Thank you so much, Dr. Alfonso. And so we come to the end of this workshop. So I would like to conclude this session, although I seriously don't want to, quit this session, because it's a really nice interactive session and informative session, especially for me. Although I was also one of the speakers, but it has acted like a eye-opener to me as well. All right. So on behalf of all the audience, all the students, I would like to thank all the speakers for sharing their knowledge and justifying to the theme of the workshop and taking out time from your busy schedules. Unfortunately, Mr. Joydeep was unable to join today, maybe because of some network issues. But I'm sure he is also going to circulate the same with the common men, like Dr. Himanshu has emphasized, that till the time it doesn't go to a normal man, because he is representative from NGO. So he is the one who is directly communicating with the people outside there. And this workshop has really highlighted on the problems, and specifically the key point, I think the good point of this workshop is that now I presume that the students or the audience, they are aware that yes, food can also be a source of something negative. But the good point is that yes, we are aware about the problem, and we also discussed about the solutions. That's the key thing, I think, because then we end at a positive note of the workshop. And I would like to thank all the organizations, Kalinga University, Milano University, and then Manipal, and then EFSA specifically. He is the head of Biocondam, so I am sure it must be very difficult for him to take out time. So once again, thank you so much. And thank you so much to our preventive team, who is also working at the back end, because these faces never come in front, but a big thank from depth of my heart. And last but not least, I would request all the audience to kindly share and spread this information to as many people as possible. And the last thing is, from my end, eat better, not less. This is one of the quote which I saw in today afternoon only. And also please join the hands together to create awareness to as many people as possible. And please do join Preventive Team to visit our webpage for more updates in this direction. So thank you so much. Keep safe, keep healthy, and stay smiling. Keep happy. Take care. Thank you so much.